ribonucleic acid. So it has the ribose sugar, but it's not missing an oxy. That difference is why the bases in DNA are A, C, T, and G, whereas the bases in RNA are A, C, U, and G. Because the U binds to the ribo, whereas the T binds to the deoxyribo. And it's going to result in a slightly different number of hydrogen bonds and all that stuff, but don't worry about it. Okay. Also, I wanted to mention this before I forget. When on your test, I asked you what kind of a molecule is DNA. That's what the Na stands for, nucleic acid. What kind of molecule is RNA? That's what the Na stands for, nucleic acid. It always amazes me. You get your final exam, the easiest test of the semester, just asking if you've got your basic information. And people forget that DNA and RNA are nucleic acids. This should be a simple question. Okay, what else is different? DNA is a double helix, right? It's 2 strands of nucleotides twisted together, right? And held together with hydrogen bonds. And the hydrogen bonds are between the bases. RNA. This is a single strand of nucleotides. Obviously, these are RNA nucleotides, and these are DNA nucleotides. RNA. It is single-stranded. It's this. But it occurs in 3 different forms. I'm going to give you simplified names for the forms because I think it's easier to visualize. The first form is linear. That's the mRNA, the working copy of the DNA. See how it's just a chain of nucleotides. What you're not seeing, here's the base, right? But it's got a little covalent bond, the sugar, and the phosphate. Little covalent bond to that one, a sugar, and a phosphate. Covalent bond, sugar, phosphate. So it really is a chain of nucleotides, just like our DNA was, okay? But we focus on the letters because that's the coding part of the DNA. That's your genetic code in there. So that's why we focus on the letters. So the linear form is mRNA, and that's a working copy of the DNA for one gene. Then there is a sort of globular form, and that one's rRNA, and a little r stands for ribosomal RNA. Ribosomes are made up of RNA and protein. What do ribosomes do? Sites of protein synthesis. So when I took my photocopy of my recipe, my mRNA, out of the office, when I went into the kitchen I actually went over to a ribosome, because that's my oven where I'm going to do my baking. Can't bake it if I don't have an oven. Gotta have ribosomes for protein synthesis. So, DNA replication happens in the nucleoid. RNA is built at the nucleoid. But when the RNA copy for the gene is done, it's literally clipped out by enzymes, leaves the nucleoid, and goes out to the cytoplasm to a ribosome. Okay? Okay, we'll come back to that when we do transcription and translation. And the third type is, I'm so sorry, this is not very professional. I call the form a turkey foot. Yeah, is there a name for that shape? The tRNA shape? There should be a name for that.
Turkey footwork?
Paper football.
What do they call it?
It's like a paper football.
A paper clip, a bent up paper clip.
Paper football.
Paper clip holder?
He said a paper football.
A paper football, oh. Yeah, I don't know what a paper football is.
The thing that would make a triangle.
Yeah, a little triangle? Doesn't look like that to me. Looks like a turkey foot. Anyway, you look at the pictures in your book, you'll decide. Turkey foot. Turkey foot. This is the way I draw them. Your book actually does them right side up. And it's got a little poochie out part somewhere.
Yeah. What the heck is that? It looks like a turkey. Yeah, Jack. That's what it looks like.
A jack, yeah,
It's
kind of
a-
like a jack. But to me it looks like a turkey foot, because usually I draw them like this. That's how I do them. That's turkey foot. See, and your book has them going in the upright direction, and I usually do them in the down direction, so mine's flipped over. Anyway, you have an amino acid at one end. I forget which end, whatever end. You have an amino acid at one end, and the other end is called an anticodon. So for this one, this would be your anticodon up here. So now you know why your book doesn't call it sense and antisense strand. Because then you have anticodons. And then there are mutations. And some mutations are missense mutations, and some are nonsense mutations. So you got sense strand, antisense strand, codon, anticodon, missense, and nonsense mutations.
Nonsense mutation is the one that finishes earlier than it's supposed to, right?
Just no amino acid. No amino acid. Okay, so that little blurb, somehow they have a little blurb in there, I just do it like that. On your anticodon end, again you have to do the complementary base pairing. So if my first mRNA codon was CAT became GUA, right? If my first mRNA codon was GUA, then that would have to be complementary base paired with an anticodon on the transfer RNA molecule that is CAU. And that would be right here. So this whole turkey foot is a chain of SP, 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 SIPI. And then these are letters. Letters are all over this thing. But the only important part is this part here, the anticodon end, the anticodon part. And this will complementary base pair the RNA. So, my RNA will be here. So what was it in RNA? It was GUA, and what was the next thing the cat was good was? What was the
It was the complementary.
GAG would be C-U-C.
Alright,
So the-
G-U-A-C-U-C-G-U-G.
G-U-G?
Mhm.
Okay. So, on a ribosome, I kind of make my ribosomes like snowmen. On a ribosome, the mRNA is going to pass through here. And when it passes through here, these transfer RNA molecules are going to complementary base pair with it. So, the first transfer RNA molecule is going to base pair with GUA. And when it does that, it's going to carry with it a certain amino acid. Remember, the amino acid was valine, right? And when that's done, this transfer RNA molecule will cleave off the valine and will leave as the next one comes in. So that's why I draw sloppy turkey feet, because it's just too much drawing. And then here will be another amino acid, which I believe was leucine, right? And the complementary to that would be GAG. What is this? If that were a T, I'd have CAD-GAG again, like my DNA. So your tRNA is going to be kind of like your DNA, except it's RNA, so it has U's instead of T's. So what happens when you're doing protein synthesis? Here's my DNA. I just copied it to RNA. Now I'm going to copy it to tRNA. I'm back where I was with my DNA, right? And now it tells me what amino acids to bring into place. And then these two will get stitched together, so we're going to make a chain of valine to leucine, and then the third one's going to come in and match up with another valine, and then those two are going to get